Okay, so now we're going to look at glucose catabolism and ATP synthesis. Glucose catabolism is the reactions involved in the breakdown of monosaccharide and use chemical energy in glucose bonds to drive ATP synthesis. Glucose catabolism has two main components, both of which generate ATP. Glycolysis, which is a series of reactions that can occur in the cytosol and splits glucose, ultimately forming an end product referred to as pyruvic acid. The citric acid cycle, which is a series of reactions that occurs in the mitochondrial matrix that helps to continue glucose's breakdown, and the substrate that feeds into that is acetyl-CoA, which will continue to break down those carbons that was part of glucose. Now, oxidative phosphorylation, this is a series of oxidation reduction reactions that take place in the inner mitochondrial membrane that allow for the use of energy liberated by the glucose catabolism. So oxidative phosphorylation is essentially what's happening at the electron transport chain. And ultimately, you'll end up producing ATP by a big enzyme complex referred to as Atp Synthase. The electron transport chain involves the transfer of electrons during oxidative phosphorylation process and ultimately leads to the production of ATP. So looking at glucose catabolism, glucose is in the cytosol. It will go through glycolysis in the cytosol, and then the end products of glycolysis will get transferred to the inner mitochondrial matrix, where the citric acid cycle will occur, and then eventually those products or those products will get transferred into the electron transport chain, which is the inner mitochondrial membrane, and you'll ultimately produce bulk of the ATPs by oxidative phosphorylation. So a great deal of potential energy is locked in bonds between atoms of glucose molecules, and the ATP can be synthesized in two ways from the energy released by glucose catabolism. The first is substrate level phosphorylation. This involves a transfer of phosphate group directly from a phosphate containing molecule, a substrate, to ADP to form ATP. This is simply a transfer reaction. It does not involve the electron transport chain, and you actually can do this under fermentative or non-oxygen or anaerobic conditions. Oxidative phosphorylation is the energy from the flow of electrons that is harnessed in the process that generates ATP. Oxidative phosphorylation is a process that occurs at the electron transport chain, and this process does require oxygen, okay? And most ATP that's produced by the breakdown of the glucose is by oxidative phosphorylation. The reactions of oxidative phosphorylation generate a great deal more ATP than glycolysis or substrate level phosphorylation. That's probably a more appropriate term to use there. And this will occur in the mitochondria as a component of the electron transport chain. Glycolysis in the citric acid cycle reduce electron carriers fed into the electron transport chain where they participate in oxidative phosphorylation. So when we say reduce electron carriers, we mean putting the electrons onto the electron carriers, which will shuttle them to the electron transport chain where they will become oxidized. Remember, reduce means they gain the electrons, right? So where they will be oxidized at the electron transport chain and then transfer those electrons to the electron transport chain, where they will undergo oxidative phosphorylation. The final electron acceptor for oxidative phosphorylation is oxygen. Okay, just keep that in the back of your head. So glucose catabolism can be divided into two broad classes, glycolytic or anaerobic catabolism, so named because these reactions take place in the absence of oxygen and involve glycolysis, or oxidative or aerobic catabolism, which is cellular respiration, so named because the reactions require oxygen. Both electron transport chain and citric acid cycles are examples of cellular respiration and refers to the cell's consumption of oxygen and the production of carbon dioxide in the water. Let's talk about glycolysis first. Glycolysis is when you take a glucose molecule and it gets broken down by a series of 10 enzymatically orchestrated reactions that take place in the cytosol of all cells. Glucose is split into two 3-carbon sugar molecules called pyruvate. The reactions then can be condensed into two phases, an energy investment phase and an energy payoff phase. During the energy investment phase, what you're going to end up doing is phosphorylating the glucose molecule on either end. So you have a first phosphorylation stage and then you have a second phosphorylation stage. Unfortunately, to add these phosphates to the glucose molecule, you need to use an ATP. And because you're adding two phosphates, you need to use two ATPs. So you need to remember that you used two ATPs to start this whole breakdown process. And that needs to be accounted for in your total net ATP gain because they were used and you don't actually have them anymore, so you have to subtract those from your total at the end of the day here. The energy investment phase, this is when you have the cleavage of the 6-carbon fructose 1,6-bisphosphate molecule, which was the molecule when we added both the phosphates, into two 3-carbon molecules. At the conclusion of this phase, two ATPs have been used and 3-carbon compounds, both of which contain phosphate groups, have been generated. And this is going to be what we call glyceraldehyde 3-phosphate is one of those 3-carbon molecules, and then diacylglycerol, which is the other one. So here we have our glucose molecule, okay? Oops, sorry, go back. We have our glucose molecule right here. Let me just throw up the little pointer so it's easier to see. Here's our glucose molecule. We had a phosphate on one end. We used one ATP. We had a phosphate on another end. Right here, we used our second ATP. And then we split this molecule right here. And by splitting this molecule, we formed two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, or right here. Unfortunately, DHAP right here, dihydroxyacetone phosphate, cannot continue down glycolysis. And what happens is it gets converted by a rearrangement to glyceraldehyde 3-phosphate. So essentially what you're going to end up having is two glyceraldehyde 3-phosphate molecules. Okay, that's why you see the two arrows here. The first arrow is forming DHAP, but then the second arrow is a rearrangement to form glyceraldehyde 3-phosphate. Normally, with DHAP, the phosphate is right here, okay? Then we go into our energy payoff phase. This is when the remaining 5 reactions are condensed into 2 steps. Phosphate groups of each of the 3-carbon sugar molecules formed by the investment phase are transferred to ADP to yield ATP. Each compound is oxidized to produce a single NADH as well. Okay, so if we look here, we're going to produce an ATP and then an ATP. Okay, this is one glyceraldehyde 3-phosphate. And then here's your second glyceraldehyde 3-phosphate. You produce an ATP and another ATP. So you're actually making 4 separate ATPs through these 5 reactions by substrate level phosphorylation. Okay, the other thing that you're going to produce is an NADH and another NADH. Those are the electron carriers. NAD is an electron carrier. It's picking up electrons that you're releasing, okay? Those electrons will get picked up by NADH. They will shuttle to the electron transport chain. When they get to the electron transport chain, NADH will release the electrons, in which will undergo oxidative phosphorylation, and then you will end up producing 3 ATPs per NADH. So we're making 2 NADHs here, right? So once we go through oxidative phosphorylation on the electron transport chain, those 2 NADHs will give you 6 ATPs by oxidative phosphorylation. Take those 6 ATPs you made by oxidative phosphorylation plus your 4 ATPs that you made by substrate level phosphorylation, and at the end of the day, glycolysis can yield you 10 ATPs. Okay. Now, don't forget we use 2 in our investment phase, so your net gain is actually only going to be 8. And at the end of the day, we're going to be left with 2 pyruvate molecules. Okay? So the overall yield of glycolysis at the end of the payoff phase is as follows. We spent 2 molecules of ATP. We synthesized 4. We synthesized 2 molecules of NADH, which will go to the electron transport chain, undergo oxidative phosphorylation, and produce 6 ATPs. We split glucose molecule to 2 3-carbon pyruvate molecules, okay? And the net yield from splitting a single glucose molecule by glycolysis is 2 molecules of ATP and 2 molecules of NADH, okay? So here, our net gain of ATPs is 2, but keep in mind, those NADHs will eventually be cashed in for ATPs, okay? And this is glycolysis in a whole. So now the end products of glycolysis is 2 pyruvate molecules. What happens to these molecules depends on the amount of oxygen that's available, okay? Under anaerobic conditions, the pyruvate is reduced to lactate, all right? And your skeletal muscles can end up doing this, all right? But what typically happens and with most cells is under aerobic conditions. The pyruvate moves into the mitochondria where it is oxidized, and then it enters into a second step of glucose metabolism, which we refer to as a citric acid cycle. This is where pyruvate loses its carbon atom, a decarboxylation step, to yield a molecule of both acetate and carbon dioxide. Acetate is then oxidized by NAD into a large coenzyme complex forming acetyl-CoA. And you essentially form 2 acetyl-CoA molecules, which will then enter into the citric acid cycle. This particular step right there, where pyruvate loses that carbon in this, what we call pyruvate decarboxylation, is an intermediate step between glycolysis and the Tca cycle. So what we have going on here is pyruvate will lose a carbon, okay? It will become acetate. Acetate will then form with CoA here, okay? And forming acetyl-CoA, all right? Now, as you go from acetate to acetyl-CoA, you are releasing electrons, which will be picked up by NAD, forming NADH, all right? And you broke off a carbon, and that's that CO2 right here, all right? Now remember, you have 2 pyruvates, so you're actually going to end up making 2 carbon dioxides and 2 NADHs, okay? And 2 acetyl-CoAs, so everything's doubled. Now what's going to happen to the NADHs? Same thing that happened on glycolysis. Those NADHs are going to shuttle those electrons to the electron transport chain, undergo oxidative phosphorylation, and because you have 2 of them, you're going to produce 6 additional ATPs. So now we enter into the Krebs cycle, the citric acid cycle, the tricarboxylic acid cycle, whatever you want to call it, Tca cycle. And this consists of 8 reactions that make up the second part of glucose metabolism and represent the beginning of the cell's oxidative glucose catabolism. This does not require oxygen directly, but it does require adequate oxygen concentrations to proceed. A small amount of ATP is produced by substrate level phosphorylation. Okay, and you're going to go through these various steps here along the Tca cycle, and you'll end up producing 10 NADH molecules, 2 FADH molecules, and 4 ATP molecules. This is from both glycolysis and Tca, okay, as well as pyruvate decarboxylation. So this is all of metabolism of glucose here. But let's look at the Tca cycle. So we have acetyl-CoA comes in. It's going to form a bond with oxoacetate. Okay, this is going to be a 4-carbon molecule forming a bond with a 2-carbon molecule, and this is going to form citrate, hence the name citric acid cycle. Citrate is going to undergo a rearrangement into isocitrate, and then you're going to go through your first decarboxylation step of the Tca cycle. Decarboxylation means we're going to remove a carbon, so we're going to chop off a carbon. So if you look here, when we do that, we go from isocitrate to alpha ketoglutarate, We lose a carbon because we broke a bond, we release electrons. Those electrons get picked up by NADH, which is our electron carrier, and we form an NADH. Alpha ketoglutarate is going to undergo the second decarboxylation step of the Tca cycle, and that's going to form succinyl-CoA. And what we're going to do is going to chop off another carbon. And when we chop off that other carbon, we're going to release CO2 because we broke a bond, we released energy, and that energy gets picked up by electron carrier forming another NADH. Okay. Now at this point right here at succinyl-CoA, we have broken down glucose completely. We have one carbon here, the second carbon here, and if you remember in the pyruvate decarboxylation step, we released a third carbon dioxide. That's 3 carbon dioxides. Remember, you have 2 pyruvates, so that's a total of 6 carbon dioxides. Glucose is a 6 carbon molecule. Glucose is gone. The problem is you can't stop right here because this is a cycle. You have to return succinyl-CoA back to the molecule you started with, which is oxoacetate. And in order to facilitate this, what's going to happen is succinyl-CoA will be converted to succinate. You're going to lose that CoA molecule, which will go back up to form acetyl-CoA. And along this process, you're going to generate an ATP by substrate level phosphorylation. Succinate then gets converted to fumarate. And instead of the electrons getting released being picked up by an NAD, the electrons are going to get picked up by an FADH. Okay? And I always remembered this because we go from succinate to fumarate. Fumarate starts with an F when we use an FADH as our electron carrier, which also starts with an F. So this is going to form an FADH molecule. FADH is an electron carrier. It will still go to the electron transport chain, release those electrons, which will undergo oxidative phosphorylation. But in this case, instead of producing 3 ATPs per NADH that we see, and FADH is only going to produce 2 ATPs by oxidative phosphorylation. Fumarate gets converted to malate, you get nothing for that. Malate gets converted back to oxoacetic acid, and you produce a final NADH molecule. Okay, so on just the cycle alone here, you're going to produce 1, 2, 3 times 2 because you have 2 acetyl-CoA, so 6 NADH molecules. 4 CO2 molecules, 1, 2 times 2 is 4. And then 2 ATP molecules, because you're going around twice, and 2 FADH molecules. Okay? That is your net gain of everything on the Tca cycle here. Because remember, you're going around this cycle twice because you have 2 acetyl-CoAs. Now going back here, each glucose molecule produces 2 acetyl-CoA molecules, which in turn turns the cycle twice. The overall yield per glucose from the beginning of glucose catabolism is a net yield of 10 NADH molecules. 2 from glycolysis, 2 from pyruvate oxidation, and 6 from the citric acid cycle. 2 FADH molecules, which are from the citric acid cycle, and 4 ATP molecules, 2 net gain from glycolysis. Remember, you make 4, but you use 2, and 2 from the citric acid cycle. Okay, so if we add this up, remember what's going to happen here is these 10 NADH molecules are going to go and go to electron transport chain, where they will undergo oxidative phosphorylation. You have 10 of them. Each one's worth 3. That's 30 ATPs. You have 2 FADH molecules, which will go to the electron transport chain, undergo oxidative phosphorylation. 2 times 2 is 4. So 30 plus 4 is 34. Plus the 4 ATP molecules is a net of 38 ATPs. Okay, by breaking down a single glucose molecule. Okay. Now, we've been talking about the electron transport chain all along the way here. And that's the process of oxidative phosphorylation. NADH and FADH get oxidized. The flow of their electrons generates an electromotive force in the electron transport chain. And what happens is with the electron transport chain, you have NAD comes in, releases the electrons, FADH comes in, releases the electrons, And now these empty electron carriers, NAD and FAD, can go back and pick up more electrons. But you're going to generate this electron gradient, and whenever you move electrons, you're also moving protons. So you're also going to generate a proton gradient. And what happens is these proton gradients will essentially fuel this big green thing here, which is ATP synthase. And this is a big enzyme complex that's going to take ADP, a phosphate, and generate ATPs. Okay? The protons are going to meet up with oxygen, and you're going to end up forming water, and the final electron acceptor is going to end up on those oxygens there. Okay. So if we calculate how many ATPs that we did this just a few minutes ago, when you add up all that, because the NADHs that you make and the FADHs that you make eventually end up at the inner mitochondrial membrane on the electron transport chain, you're going to end up making ATPs. So that gives you a total of 38 ATPs that we talked about, that you can see here. Okay. Now the problem that we're also going to run into for a second here is that, and some textbooks don't actually account for this. Most textbooks just say you make 34 to 36 ATPs. You're making 38 as a net, okay? You're actually making 40, but we use 2 here in glycolysis. The other area where you're going to use ATPs is the transport of pyruvate against this, through this outer mitochondrial membrane and this inner mitochondrial membrane. Pyruvate's a large molecule. It's not going to be able to flow right through, okay? And what you have to do is you have to go through a transporter, outer mitochondrial membrane, TOMM, which is TOM, and transporter inner mitochondrial membrane, which is TIMM, or TIM. So you have to go through TOM and TIM, and those are ATP-dependent transporters. And you're actually going to use 2 ATPs there as well. So if we subtract those 2 ATPs, our net total in our cells is going to be about 36. Okay. All right. And that's it for glucose catabolism. All right. I know some of you probably have gone through these pathways more than you care to, and this is hopefully going to be a review for you. But if these aren't pathways that you guys have seen before, you know, please feel free to reach out and we can walk through this step by step. Okay, especially my physics guys. You might not. I don't know. I'm not sure if you guys have done these before.